Hey guys, so I'm going to go a little science nerd on you and talk about something called CRISPR. Now, before I really go into what CRISPR is, I need to explain a little bit about DNA. So for those of you that don't know, DNA has a double helix structure that is made up of billions of nucleotide pairs that are joined by hydrogen bonds. And these four nucleotide bases are adenine, thymine, cytosine, and guanine, A, T, C, and G. Now, in DNA, A always binds to T, adenine to thymine, and C always binds to G, cytosine to guanine. Never any other combination. This could end up in a really bad mutation that would probably not end well for us. So, now that we know a little bit about DNA, let's move on to CRISPR. Now, CRISPR is short for clustered, regularly interspaced short palindromic repeats. And the reason they're called palindromic repeats is that if you line up the repeated patterns of A, T, C, and G, they read the same backwards and forwards, like palindromes in the English language, like mom or dad or even taco cat. Now, as we know, every living thing on Earth has DNA, from us to the biggest elephant or blue whale to the teeniest, tiniest bacteria. And this is actually really cool. We actually found CRISPR in a teeny tiny bacteria called E. coli, where we get a lot of other really cool genetic things from. So, on its surface, when we first look at CRISPR, it seems a little counterintuitive. So, CRISPR is these little splices of DNA inside the regular DNA of a bacteria's genome. And the reason this is counterintuitive is because these splices of DNA actually come from a bacteria's worst enemy, a bacteriophage. Now, the reason a bacteriophage is so bad is because when it attaches to a bacteria, it injects its own DNA into the bacterial cell, which can essentially turn an unprotected bacterial cell into a mindless bacteriophage-producing zombie. And when it turns out the bacteriophages, this kills the cell. So, why would it be beneficial for a bacteria to have bacteriophage DNA in its genome? Well, bacteria with bacteriophage DNA in their genomes actually have a little bit of an advantage. Because what CRISPR does is when the bacteriophage injects its DNA into the bacterial cell, it pairs up with something called Cas, or CRISPR-associated proteins. And what happens is the bacteria churns out these CRISPR-associated proteins, or I'm going to be referring to them as Cas from now on, and they take the bacteriophage DNA, or CRISPR, with them. And when they encounter similar bacteriophage DNA floating around in the cytoplasm, they actually latch onto it, breaking it down and destroying it, thus saving the cell. Now, you may be saying, well, there are millions, possibly even billions of bacteriophages out there. What happens if the bacterial cell encounters one with DNA it's never seen before? Now, this is the really cool part. When a bacteria encounters a bacteriophage with DNA it's never seen before, it will continue to churn out the Cas proteins. But instead of taking the CRISPR DNA with them, they will latch on to the new bacteriophage DNA and break it down. But now this is the really, really cool part. They actually insert pieces of the bacteriophage DNA into the already existing bacteria's genome, so that when the bacteria encounters bacteriophages with similar DNA in the future, they'll be better protected. You know, it's sort of similar to why we get vaccines when we're little, so that our bodies know how to fight them when we get older. Now, this can actually have big possibilities in humans, because we have a lot of genetic diseases that could possibly be fixed by CRISPR. Possibly. They haven't really figured out how to do this in humans yet, But scientists are currently working on a way that we could use CRISPR to fix genetic diseases such as Huntington's or sickle cell anemia. Now, the way this would work is that they would feed the Cas proteins splices of DNA from someone with Huntington's or sickle cell, and they would find the particular genes that cause these genetic diseases and give them to the Cas proteins. So then they would inject the Cas protein with the CRISPR bad genes into the person's cells. And what would happen, theoretically, is that when the Cas protein encounters DNA that matches the bad genes that it's carrying, it will remove the bad genes from the human's DNA, thus allowing the DNA either A, to repair itself, or B, for the scientists to be able to introduce new, healthy genes into the place where the bad genes were. Now, this could have really big repercussions for us, because this could possibly lead to a future without genetic diseases. I don't personally know how I feel about that. I'm all for curing genetic diseases, but sometimes for me it kind of feels like we're trying to play God. But really, I think I just figured out how I feel about this. If we can help people with diseases that can't be cured any other way, I think we should do it. Now, all this research is really theoretical at this point. They really haven't figured out how to do it in humans. They've done it in non-viable embryos. Embryos that were really not gonna die anyway. But the ethics of it haven't really gotten fixed in the scientific community yet. So, I hope we can figure out how to do this someday in the future, because I don't know about you, but someday I would love to live in a future without genetic diseases. Well, that's all for today, guys. I'll see you later.